First, let me remind everyone that you can claim CME credits. The code is at the bottom. You just need to sign up with the CME group, and once you text that, you can get credits. I think typically we'll put it in the chat as well, so you can access it there. And also encourage everyone to follow us on social media at the bottom. Dr. Jamie Kroon's done an excellent job in getting that up and running and being fairly active. So I'm very pleased today to have a presentation from Dr. Anika Guha. She'll be talking about lifetime cannabis use and its association with brain volume and cognitive functioning in middle-aged and older adults. Dr. Guha's research examines the effects of cannabinoids and psychedelics on neurocognition and mental health across the lifespan, with a particular focus on adolescents and older adulthood. She previously worked with Dr. Bo Anschutz at Washington University in St. Louis. So Bo is well known to us here in the HIV group for decades. She received her Ph.D. in clinical psychology from UCLA and completed postdoctoral training at the University of Colorado Anschutz Medical Campus, where she focused on translational neuroimaging research. Dr. Guha currently works in the Cannabis, Health, and Addiction Over the Lifespan, or the CHAOS lab. The CHAOS lab is directed by Dr. Kent Hutchison, who's a professor of psychiatry in the School of Medicine at the University of Colorado. He also directs a NIDA-funded Center on Excellence to study the effects of cannabis over the lifespan, the Rocky Mountain Cannabis Research Center. Dr. Hutchinson is also on the line today to address any additional questions that perhaps come up after Dr. Guha's presentation. So Anika, I turn it over to you, and thank you again for doing this for us.
All right, well, thank you so much for that introduction. Hopefully, can everyone see my slides?
We can.
Great. So yes, I will be primarily talking about some recent work, a project we did utilizing data from the UK Biobank to look at associations between lifetime cannabis use and brain health in an aging cohort. But I'll also briefly discuss some prior work by our group, most of which occurred before I joined the group, which is why Kent being here is super helpful if any specific questions come up. But I'll talk a little bit about some of those projects that kind of led us to this project, and then talk about this UK Biobank project, which has gained some attention in the media because people love to hear about, is cannabis good for you or not? So go ahead and get started here. So we know that cannabis use is rising in older adults. It's more widely available. People love to tout its health benefits. And so we've seen huge increases in use in this demographic, tenfold between 2007 and 2018. We also know that older folks tend to use cannabis for different reasons than younger folks, often citing health reasons, so wanting to address issues, such as chronic pain, sleep issues, and anxiety. And not only do we have more older adults using cannabis, but we also have a demographic of aging cannabis users. And we don't have a ton of great data on what the long-term effects on the brain are in this demographic. And so for a lot of reasons, we just really need to better understand these relationships between cannabis use, brain health, and cognition among older adults. And as you can see on the cover of this magazine, as I said, cannabis is touted for having various health benefits. And so we're talking about aging, we're talking about brain health. So it makes sense to then also consider, well, what are the impacts on dementia and dementia risk? Are there benefits when it comes to cannabis? Are there possible adverse effects when it comes to dementia risk? And that's really important for us to know, especially as we have increasing dementia rates because our population is aging. And obviously, there's such a huge economic burden as well. So when we think about what's been done and what is out there when it comes to the effects of cannabis on the brain, and I'm sure everyone here has read many studies, the kind of long and short of it is that the results are varied. And actually, a lot of research has tended to focus on the negative effects of cannabis, including reductions in brain volume associated. But of note, a lot of that work tends to focus on adolescents and young adults. So we tend to see kind of that negative portrayal of cannabis during this sensitive period of brain development. That said, there was a recent paper from 2024 that similarly utilized the UK Biobank and actually did find overall reduction in gray matter volume among cannabis users. But I'll just want to highlight that they really just looked at overall total gray matter volume. There wasn't sort of any specificity when it came to what parts of the brain might be impacted. And when it comes to cognitive impairment, again, a lot of the literature tends to focus on adverse effects, but also there tends to be a focus on acute effects. So often, what is the short-term impact of cannabis use right after taking it? And so not only is there a gap on understanding of the impact in older adults, but also long-term impacts on cognition. So in contrast to a lot of the adverse effects that we tend to hear about the most, there is a lot of kind of upcoming preliminary evidence to suggest that there could be benefits to cannabis use in older adults. Um, and so we know that one thing that might separate out when we see benefits versus adverse consequences, for example, highlighting that UK Biobank study where they found reductions in overall gray matter volume, it may be important to look at specific parts of the brain. So cannabinoid 1 or CB1 receptors, which are the cannabinoid receptors most populated in the central nervous system, they are expressed widely, but there are particular regions that have a higher density of these receptors, including several subcortical regions. So it may be really important to look at specific parts of the brain, especially because there may be parts of the brain that, we think that cannabis is acting, more specifically on based on cannabinoid receptor density. Additionally, the animal literature has really demonstrated some promising mechanistic actions of cannabis when it comes to aging, a possible neuroprotective effects that could be related to its impact on inflammatory pathways, as well as roles in neurogenesis and brain-derived neurotrophic factor. And so we have good reason to believe that there could be reasons why cannabis is good in the aging brain. Another consideration when we think about why cannabis effects might be different in older adults versus young adults, we know that the endocannabinoid system changes as we age. And specifically, we know, for example, that the number of our CB1 receptors peaks in adolescence and then declines pretty sharply as we age. And the loss of those receptors is associated with age-related changes in the brain, including loss of hippocampal neurons. And the hippocampus is obviously such an important region when we think about brain health and cognitive function. It's also one of the most susceptible to age-related decline in terms of volume, and it is obviously plays a role in dementia and dementia risk. And so there's actually some really interesting animal model research that has shown that, again, really distinguishing the effects in younger and older individuals. When we look at animals, THC in low doses seems to reverse cognitive decline among aging animals, but we actually see a harm effect or a negative consequence in young animals. So again, it really appears that age is an important moderating factor when we look at effects on the brain and something we really need to be considering in our work. Just to highlight some of the kind of big questions that we've had as a group. Clearly, the physiology is different when we think about younger versus older adults. So the risks and benefits might be different, and it's important for us to know. The acute effects might be different in a variety of domains, including cognition, impacts and things that are really relevant to aging, like balance, as well as the long-term effects. And that really is where we think about things like cognitive function and brain structure and morphology, as well as some of the areas that, again, older folks might be using cannabis for, including dementia, pain, and sleep. So I'll just give a little bit of info from some prior work in the group that kind of led us to our work with the UK Biobank data. This first study, it was a study of acute effects of THC, THC plus CBD, and CBD only products to look at differences. And although the study wasn't specifically designed to look at aging specifically, we did separate out the participants in the study into a younger group and an older group so we could look at moderating effects of age. And although prior work by our group hadn't been finding main effects of THC on cognition, we were curious if there might be a moderating effect of age. This first plot is showing differences in plasma THC levels following administration of these products. The gray bars are CBD only product, the pink bars are THC only, and the green bars are the combination. And just to orient you on the on the x-axis. The leftmost bars are going to be the time point 0, which is right after administration. And then the rightmost bars of a color will be 60 minutes out. And then within each pair of bars, left is the younger and right is the older. And what we found when we looked at these THC plasma levels is that the younger group showed significant differences in THC plasma between CBD and THC and CBD, where the THC only group had the highest levels, but we didn't see this difference in effect when we looked at older adults. And then specifically looking at effects on cognition. So again, this is again thinking about acute cognitive effects. What we saw was that older adults showed better cognitive performance than younger adults, both at that 0 point and 60 minutes out. So the leftmost plot here is episodic memory, and the right is processing speed. And specifically in that THC group, what you're seeing is that the older adults are showing better performance than the younger adults. So really highlighting that even in the acute phase of use, we're seeing differences in response in our older adults. As I mentioned, the endocannabinoid system differs in younger versus older adults. So this is data from studies that were looking at endocannabinoid levels. This is something we really like to include in our work. So we collect plasma endocannabinoids, and we can look at differences in those endocannabinoids at baseline, before any use of cannabis product, and then after. And so this is data that was part of a secondary analysis from 2 different studies of regular cannabis users. And this allowed us to create 3 age groups: younger, midlife, and older participants. Participants, because they were regular users, were asked to abstain from using cannabis prior to their baseline draw, and then differences in endocannabinoids were assessed pre versus post use. And so to orient you to these plots, the purple bars are the young group, the blue are the middle-aged, and the green are the older group. And these are showing baseline levels of different endocannabinoids. On the left is anandamide, in the middle we have DEA, on the right we have LEA. And what we found is that even at baseline, there are differences where the older adults are showing lower endocannabinoid levels than the younger adults. Then switching gears to response to cannabis, what we see is that the older adults also show the largest change, the largest increase after using cannabis. Specifically, we would look at anandamide on the left and DEA on the right. So you can see again that green bar is the older adults, and they're showing a larger increase. So suggesting that older adults not only show differences at baseline when it comes to endocannabinoids, but also differences in endocannabinoid responsivity. Some data from a study that was specifically oriented to older adults. This was comparing regular users to controls, so never users. And you can see that the mean use was around 20 years in the regular users. So this is pretty consistent use of cannabis. And this specifically focused on differences in brain morphology as well as cognitive performance. This plot is showing regional brain volume separated by left and right hemisphere. So each set of bars on the x-axis is a brain volume, where again y-axis is the actual volume, and the y-axis is the volume, and the x-axis is showing those different regions. What we found was that 3 regions showed significant differences, wherein the cannabis users had larger volumes for left pallidum and bilateral putamen. Only the left putamen withstood FDR correction. But it is worth noting that for the regions that are showing a difference, we're seeing larger volume among the cannabis users, and there were no regions where we saw the opposite pattern. So we weren't seeing any obvious deleterious effects. And this is just to show a little bit more information about those cannabis users. So just to kind of separate out, if you see on the right side, this is kind of distinguishing those cannabis users by sort of more lifetime use on the right and the short-term users on the left. So you can see they differ in their years of regular use, 4 years versus 40 on average. But in general, I think it's worth noting that these folks are reporting a pretty high level of THC use. So on the TLFB, you're seeing average of 54 or 71 days of marijuana use, and pretty high amounts of THC on the bottom in terms of milligrams. So even in this context of pretty significant THC use, we didn't see any obvious sort of negative associations. The last previous work I'll focus on comes from a secondary analysis of a study that was looking at relationships between exercise, the age, and exercise age and inflammation, and specifically focused on brain volume and cognitive function. These were our older adults, and there were a subset of those folks who reported regular or weekly cannabis use. And really, I think this highlights sort of a replication that when we are seeing differences in older adults, there tends to be larger regional brain volumes, especially for, you'll see similar regions here from before. You'll see putamen and pallidum on the bottom here, as well as caudate and hippocampus. But generally, what you see is the non-users on the left are showing smaller volumes than the users on the right. So again, when we look at these subcortical regions, again, generally understood as having pretty large, pretty high CB1 receptor density, we're seeing larger volumes among cannabis users when we look at older adults. So that brings us to the UK Biobank study. We really wanted to expand on this work and extend it to a larger sample. As you saw, a lot of those studies are relatively small samples of older adults. So the UK Biobank, which I'll talk about a little bit more in the next slide, just provides us a really nice large sample of older aging adults. With the hope that, kind of having that power, that large sample size would enable us to look at some of these relationships more clearly. And so we looked at specifically associations between lifetime cannabis use, brain volume, and cognitive functioning. For those not familiar with the UK Biobank, this is a very large prospective cohort study of over 500,000 individuals. They were all recruited between the ages of 40 and 69 in the United Kingdom between 2006 and 2010. So it's this very large, rich data set. They collected a lot of different measures, including neuroimaging and cognitive assessment. So always nice when you can avoid paying for tons of neuroimaging yourself. And so you're capitalizing again on this huge data set. The consequence of that is it was not specifically designed to assess cannabis. So the questions that they ask about cannabis use are relatively rudimentary. We don't have any measures of things like endocannabinoids. So again, the pros and cons of this data set are really large sample of aging adults. The downside is we are a little bit limited in what we can ask when it comes to cannabis use. Based on the prior work I showed you, we predicted that adults in the sample who reported lifetime cannabis use would demonstrate greater regional brain volumes in those subcortical areas that we tend to see those effects that we saw earlier, specifically regions that have that high density of CB1 receptors. And then when it came to cognition, we were still a little unclear what to expect. We had that data showing that older folks tended to have better cognitive performance acutely, but it's still unclear in terms of long-term relationships. And again, a lot of the work in younger adults has tended to find or report negative effects. So we weren't totally sure what to expect when it came to cognitive function. Here is the primary question that folks in the study were asked about cannabis. Have you used cannabis even if it was a long time ago? So pretty broad question. Folks responded in one of these categories: no, 1 to 2 times, 3 to 10 times, 11 to 100 times, or more than 100 times. We figured since we're already having to work on a categorical level, we don't have that full continuum. We decided to sort of collapse into 3 categories to generally get at non-users, low to moderate users, and then sort of higher users. So the moderate group was 1 to 100 times, and the high use group was more than 100 times. And so that was kind of our way to try to get a little bit at dose-dependent relationships. And then we didn't have great access to data about, what was your use in the past month or the past 2 weeks, or do you currently use? But they do ask a question, which was age when last took cannabis. And so we decided from that question, what could be interesting is to specifically highlight folks whose last use or only use really was before the age of 25. So really saying, okay, these are folks who use during that sensitive, vulnerable developmental period that we think is really important. They haven't been using since then, according to their own report. Do you see differences between those folks and non-users? Are there sort of long-term possible effects of that adolescent use? So yes, as I mentioned, these folks we obtained they obtained structural and functional imaging. We focus on this project on structural. We have another project looking at some functional connectivity analyses. But here we specifically wanted to look at a priori regions, again, that we had seen in our own work, as well as some other studies specifically looking at regions with that high density of CB1 receptors. And that included the putamen, hippocampus regions. Regions that you saw in some of those previous slides. And then we focused on cognitive assessments that hit some of those domains that we've seen linked to cannabis use before, learning, processing speed, memory, and attention. And again, from our acute work, we had seen processing speed and memory come up specifically in terms of places where we saw differences in our older versus younger participants. A big question when you have such a big data set and it's not an RCT. So what all do you include in your analyses? And I think this is something that we had to really think about mindfully. You can quote unquote control for a lot of different things, but then it can maybe reduce generalizability and cause overfitting. So we really wanted to be thoughtful about what we included in our models. Of interest, we did include sex as a factor because we know that men and women tend to use cannabis differently, different subjective responses as well as physiological responses. And the endocannabinoid system does seem to interact with hormones. We see sex differences in things like CB1 receptor density. So I really wanted to make sure that we were including sex in our models. And then you might be surprised to see that age is listed as a covariate of non-interest. The reason for that is we initially looked at age interactions because we figured those would be especially important, but we actually didn't see any significant effects when we looked at those interactions, likely because we already had a constrained age range. So instead we had age in as a covariate of non-interest, just to continue to control for possible age effects between our groups. And then we also wanted to control for the confounds of polysubstance use. So we included smoking and alcohol as well, especially when you're looking at Europe data. The co-use of tobacco and cannabis is very common. We also know that cannabis users tend to use other substances like alcohol. So we really want to make sure that we were trying to distill out cannabis-specific effects as much as possible. And then we also included ICV in our MRI analyses to account for inter-individual variability and to really help us get at some of those region-specific effects. Here's just a quick table showing those groups. May not be surprising, but the largest group was the non-users, so 20,000 folks in that group. And then we have the moderate use group, then is smaller at 5,000. And then the smallest group is our high use group, only 633 folks who had both imaging and cognitive function data. There were significant differences for all those covariates I mentioned. So following generally linear patterns. So as you move up in terms of cannabis use, you see participants are younger, they tend more male, higher reports of smoking and alcohol use. Starting with some of the main effects of cannabis use on regional brain volume. So these plots are all showing brain regions that showed a significant main effect of cannabis use. With, again, the left side is the non-users, the middle is the moderate users, and the right is the high users. So to highlight the boxes in red, these are all regions where the moderate use group showed significantly larger regional volume than the non-use group. And that includes bilateral putamen, bilateral hippocampus, left amygdala, and right caudate. Highlighting now that red box for right amygdala, you see that this was a region where actually the high use group had significantly larger volume than the non-use group. I also put just these dotted red boxes just show numerically regions where that high use group seemed to have the highest brain volumes, even though it was not significant with pairwise comparison, which may mean that it's not a real effect. It could also mean that we're really dealing with variance issues, since you can see the error bars are much larger for that group because it is much smaller than the other two. And then lastly, highlighting that not all these regions showing an effect showed largest volumes in the cannabis users. For bilateral posterior cingulate, we actually saw that the largest volumes were the non-users. For left compared to the high use group, and for right compared to the moderate use group. So again, I think it really highlights it's important to think about where in the brain we're looking for these effects. It's not an all or nothing situation. And although we did do correlations to look at general relationships between brain volume and cognitive function, generally thinking larger brain volumes in aging are a good thing because we know that when our age, we have degeneration and atrophy. It's not unilateral. So, there are some studies that suggest that posterior cingulate, this hub of the default mode network, larger volumes may not be a better thing. So, again, we don't know 100% what the functional impact of this is. But again, I do think it just makes it really important that we, don't look too broadly. We really need to take a little bit more of a nuanced approach. Now, this is specifically looking at those folks who used in adolescents only compared to the non-users. And this is, again, still looking at brain volume. So on the left is non-users, and on the right is adolescent users. So again, folks had not used since they were 25 or younger. And we saw for all the regions showing a main effect. For all these regions, the adolescent users have higher volumes than the non-users. And you can see, again, bilateral putamen, bilateral hippocampus, left amygdala, and here you see right accumbens. Switching gears to the cognitive measures just, again, I know folks here know about cognitive function measures, but just as a reminder that for most of these, higher bars are a good thing, better performance. But we do have, for example, in the top right, trail making, number of errors, so lower is better. And same with the bottom left and middle bar graphs, which are showing duration to complete or reaction time measures. And what we saw is that for all the cognitive assessments that showed a main effect of cannabis use, we saw better performance among the cannabis users compared to non-users. And typically we're seeing that difference is most significant between the non-users and moderate users, although for numeric memory impaired associate learning, we're also seeing a difference between non-users and heavy users. And again, splitting it up then between the non-users and adolescent users, again, adolescent users are showing consistently better performance than the non-users. This slide is showing all the cannabis by sex interactions. And it's complicated, I think, is the big picture. It wasn't consistent that, men or women always had better effects or consequences of cannabis use. It was inconsistent across measure. And so I think that for me, what this really highlights is we need to continue to understand sex as a moderating factor when we think about effects of cannabis on the aging brain. And I think a really good indicator of that is this bottom right plot of prospective memory, where you see a very classic, X interaction where let me see if I can get my mouse here, where you can see that non-using females had better performance than the adolescent using females, and the exact opposite was true for men. So I think, again, this just really highlights that we need to be keeping sex in mind when we look at cannabis effects on the brain and cognitive function. So some of the takeaways, I think that utilizing this very nice large sample from the UK Biobank, we're really getting some additional support from our earlier findings that there may be benefits to cannabis in the aging brain. And again, that this really comes at odds with a lot of the literature that has generally focused on adolescence and these detrimental effects of cannabis in adolescence. I think it also highlights that those relationships are nuanced and complex, even within older adults, and that they likely depend on a lot of different factors, some of which we had some data on, and some which we didn't because the UK Biobank wasn't designed to study cannabis. So clearly, the developmental stage, when people use, and also when we're assessing them, likely matters. The regions we look at and the types of cognitive function we look at matter. The dosage matters. We were able to see some differences when we think about moderate versus high dosage, but again, more detailed measures would be very helpful for that. And then types of cannabis. So considering most of these folks were reporting cannabis use several years ago, that was probably, smoking a joint, flower. But nowadays we have concentrates, we have edibles, we have vaping. So that likely makes a big difference. And that will continue to be important considering the current landscape of cannabis use here in the US. And then, as I mentioned on the last slide, just really the importance of evaluating sex effects. Clearly, that makes a difference when we're looking at these relationships. So this is just highlighting some of the takeaways for us in terms of how this informs our work or impacts our work. Again, we really want more specific cannabis measures, and that's what we are able to do that since we're collecting our own data. For example, as I mentioned, dose dependence is likely very important. We think that there's likely an inverted U shape when we think about the relationship with cannabis and a lot of these brain measures, but we need sufficient measures to determine that relationship. We also really like to have those measures of biomarkers, so understanding the role of endocannabinoids, how those change, especially with aging, as well as knowing what products people are using. THC, CBD, those we know have very different effects. And then ideally having longitudinal design so we can look at changes over time. So the next step for us, as I alluded to on that first slide, we're talking about aging, we're talking about brain health. So how does this impact dementia risk? Again, this is such a huge public health concern. And so I recently submitted a grant to really look at this. So again, our preliminary data suggests there are these benefits to cognitive function, to brain volume, that there may be, again, based especially on the animal literature, mechanistic pathways that suggest THC could mitigate age-related cognitive decline, which may relate to risk for dementia. And again, this may be mediated by things like inflammatory pathways, but we don't have a clear picture of that yet. So that's a direction we want to go. And additionally, again, we think that maybe low to moderate doses of THC may be beneficial, but we think it's possible that high doses of THC could be harmful. And we need better data to clarify that as well. So in this project, we plan to compare regular users to never users between the ages of 60 and 75. And we're going to have a large swath of measures so we can really clarify some of these important relationships and ideally help to identify mechanisms of action, including measures of cognitive function, structural and functional brain imaging, risk factors such as neurofilament light, endocannabinoids, inflammatory markers, to really help us clarify the picture. And again, the idea being that if we can better understand what's going on, we can really help to understand, is cannabis helpful? Is it harmful? Does it depend on the dose? These are all important questions. So this is just an overview of the aims of that project. First, just looking at the effects of cannabis use on all those measures I mentioned, then looking at whether endocannabinoids or inflammatory markers mediate those effects. And lastly, is sex a moderator of those effects? Because again, we've really seen that seems to be very important. And then I just wanted to give a quick highlight that we're also extending this work to the psilocybin space. Again, because we're seeing increasing rates of psilocybin among older adults. And so we have recently submitted a grant with a very similar structure to the one I just described, but specifically looking at psilocybin use in this demographic. And I think this is really interesting, especially for those of us in the cannabis research space, because psilocybin is really following in the footsteps of cannabis when it comes to things like legalization and what we're seeing in the marketplace. We're seeing that more older folks are using psilocybin, often for health effects. This is definitely clear here in Colorado, where it's been decriminalized. We also see in the animal model literature that it influences a lot of those similar age-related pathways, inflammatory pathways, neurogenesis pathways. So again, likely has some impact on brain health over the lifespan. Um, and also that those effects likely different over the lifespan. For example, metabolism, a lot of the components of psilocybin is very different in older adults compared to younger adults. And lastly, again, we just don't know, is it good? Is it bad? Is it helpful? Very unclear as of yet. And so we need more data to help us clarify those relationships as people continue to use these products more and more. So with that, just want to thank all the participants who participated in the UK Biobank so we could do this project. All our colleagues and collaborators through the Rocky Mountain Cannabis Research Center and the Trend Center as they helped us to access the UK Biobank data. So thank you.